All right. Hello, everybody. Phitonex webinar work from home edition, probably for everybody. Hey, Richard, how you doing? Great. See some familiar names since I can't see familiar faces on the list here. I'll tell you what, we're going to give it 2 minutes. And as much as I'm sure you want to stare at me, I'll stop my video for those 2 minutes. And then we'll get started at about 10:02. So if you need to grab some extra coffee, get ready for just talking about lasers, because I will not be mentioning anything else other than lasers and fluorophores for the next hour, which will probably be a great distraction, and then hopefully educational. So anyway, we'll get started at 10:02, and look forward to reconnecting on that in about 90 seconds. All right, let's do it. Cool. All right, here we go. I'm not going to mention the virus. We're not going to do it. We're only going to talk about fluorophores. We're going to talk about wavelengths. We're going to talk about dyes, because that's really what's fun and it keeps us focused. So anyway, if you've seen our previous webinars, there's only going to be a few updates here, but I really want to get into the technology, some of the status on some of the large experiments that we're doing right now, and also talk about some of the things customers have been up to with our fluorescent labels. So happy you could join us today. Let's have some fun. And there's a Q&A function. I'll answer those live in session as those come in, particularly once I can get up and out of the PowerPoint. Cool. So I always like to start with this question. It's maybe my most favorite question now, which is, what if you could design a fluorescent label from scratch? What if you could just start over and throw out some of the labels that we've been using for a long time? What would that mean for panel design? How would you think about that in the context of this? What would it mean in terms of the way we could leverage our instruments, the ones we currently have and the ones that are being developed? And I think there's three things. There's three things that we'll build out today, but then we'll talk about the current labels and sort of how they stack up into this environment. The first one is pretty obvious. It's the one that really comes to mind the most quickly, which is, Mike, they would be bright and clean, right? We would try to make them silent in the violet and the UV, and then increasingly even the deep UV, right? This has been a bit of a dumping ground for spread. And regardless of our ability to distinguish signatures, if we're detecting more spread than signal in the violet, we've got kind of an issue, right? And it also leads to underutilization of the other dyes we can use in a multicolor panel. The other part is somewhat obvious, but it's something that we've been dealing with for so long with APC and P that we've just sort of gotten used to the pain, right? Which is limiting cross-laser excitation. So we'll talk about that and how we specifically design for that. Consistent and stable, this is something that's, I think, very interesting. It's emergent for us, for sure, in terms of what's the stability of your fluorophores and how could you make something that's really, really stable over time, or in fixative solution, right? The other one is this notion around controllable brightness, right? What if you could choose your brightness? Instead of just dealing with, hey, we've got P or P conjugates really bright, right? And sort of like my favorite quote from a call yesterday with a customer was, everybody hates P side 5. And I don't think we hate it. I just think we have to use some of these fluorophores sometimes to get the biology done. So we'll skip past this, but getting past into panel design, when you think about panel design, all the things you have to bake in to creating a panel, right? You've got spectral overlaps, you're looking at spectral viewers, you've got this underlying notion of antigen density, which you then have to map up with brighter fluorophores. You have spillover spreading, which doesn't go away when we move into, for instance, we move from traditional to spectral flow cytometry, right? There's still spread, there's no magic around that. Still makes the data very hard to analyze. And we also got to worry about staining index or separation index if we want to take into account the difference in between the negative and the positive and the width of the underlying peaks. And really, if you think about it, underlying dye performance affects really 3 of the 4 of these, right? Because it's certainly not affecting the underlying antigen density. And so we end up dealing with compromises on 3 of the 4 of these items. But with respect to antigen density, it's actually kind of interesting because I think dye performance certainly affects our ability to multiplex when you have multiple low antigen density markers, right? So imagine looking at, for instance, multiple co-inhibitory or co-stimulatory markers at the same time on cells off even adjacent lasers. That sounds essentially impossible with the current dyes that we're using in the laboratory, but I think becomes something that's possible and something we're investigating with customers as I'll share with you. So let's look at all the 7 available laser lines. Like if we built an instrument, it had 7 lasers, right? This is where we would focus it. There's really not much going on right now off the deep UV. There's really none to date. Although I'll mention that a lot of the UV dyes very likely are putting, at least from what we've seen, are putting a lot of signal into that as you'd expect, a lot of spread in the deep UV. IR, there's really only this zombie near IR dye, not a lot of work in the IR. And then we deal with cross excitation and spread really across UV, violet, blue, yellow, green lasers, and red. And really where we focus our attention has really been into that blue, yellow, green section, right? And so if you look at, well, what's being cross excited by what? So what I've got here is on the y-axis, I've got the primary laser, right? Something that's being excited, right? For instance, off the blue or the yellow, green. And then I've got the cross exciting laser. And so, you sort of end up in this minefield when you're thinking about looking at all of these different spectra of how am I going to, what this going to look like on a cross excitation basis, right? And you have labels, particularly in the BYG, where you've got PE or D tomato that's that should be right off the blue, being cross excited by the blue laser, and then you've got blue labels like per-CP being cross excited by the yellow or green, right? And you just end up in this matrix of compromises. Again, we focused in particularly on this BYG line, thinking about what we could do to move away from things that have, that might be very, very bright, right? Whether it be PE or some of the blue dyes, but can't be used off adjacent detectors and really unlocking current instrumentations, be they something like an LSR or an LSR Fortessa or a Cytoflex, or be it something like an Aurora or a Symphony, right? Really across that spectrum, how do we create plug and play dyes where you can immediately get more out of the BYG part of the spectrum, right? And so we've, looking at some really nice work here from Cytek and looking at the performance of current dyes. This is something that we've really thought a lot about, and I think it's been really kind of brought to mind by leveraging these instruments is, what does it look like, right? What do these labels actually look like, right? So this is this is looking at a dye, and if you're looking at these plots, these ribbon plots, you've got the primary detector here with this band, and then everywhere else, it might be a signature, but it's also introducing spread. So regardless of the ability to distinguish these, you're still having a really tough time with data analysis if you're introducing huge amounts of spread, or you're plowing more and more spread into these channels that are off the primary detector, right? So in looking at this, and they've done a great job in looking at a multitude of different dyes here. You've got per-CP, Brilliant Blue, and BB700, and you can just see that there's there's areas where these labels are adding an enormous amount of spread. In particular area that we're sensitive to is in leveraging the violet, right? When people are using these violet dyes, the Brilliant Violet dyes, and have really built a lot in multiplexing capability for these violet dyes, but there's so much noise being driven in from, the blue and the yellow green excited dyes that you really end up with a tough data analysis situation. And really, you end up in this place where you think about panel design, you just end up with a ton of compromises as you map in what we're handed essentially from brightness. And really a lot of these have been handed, they've been dyes that have been with us for more than 15, 20 years, right? And so we map in and we say, okay, the lowest antigen density is going to get the brightest marker, right? And it's even interesting when we think about brightness. What does brightness really mean? I think this is something we'll come back to, right? Where brightness really should mean the ability to separate populations. So how do we really enable that? Okay, so let's go back to our requirements, right? 3 things, very simple: bright and clean, consistent and stable, and controllable brightness. So how do you actually design for that? And so the platform that we have is we've got single-stranded oligonucleotides. We bind these onto single component organic dyes, quite small. And what we can do is anneal them into this phyton structure. And the phyton structure is an artificial structure. It has this cruciform shape. And each one of these arms we call a cluster. And what that enables us to do, right, is we can perform the sequence design and we can engineer down to the sub-nanometer level the way that, for instance, dyes might interact with one another in a fret. If you think about, so if you remember your fret equations, and who doesn't, it's proportional to the square of distance. And what's very, very interesting is that you can actually tune that, right? You can tune the amount of fret that you get. You can tune actually the efficiency of fret energy transfer that you get. And it might actually be different for different donors, different acceptors, and it might be different for different lasers, right? So if you have a blue laser, you might actually say, okay, given that blue laser in this donor, we can actually tune that fret. And that's exactly what this platform enables us to do on platform, right? So these are not fret dyes. There's actually, that's what's occurring on platform. And you'll see how that manifests when we look at the excitation and emission profiles of the 6 labels, right? The other thing that's quite interesting, and this is what gives us for its worth tunable excitation and emission, but the other thing that's quite interesting is because we have this platform, we can use each one of these clusters to tune the brightness up and down as if we have a volume knob, right? And that becomes interesting. So I'll share some of the work that's in progress in leveraging that tunable brightness capability and the way that we're thinking about it, right? And I think this is an area that's pretty exciting. And what we can do is then take, put a single-stranded oligo off the end of this phyton and then conjugate it to an antibody. And this is actually the way that we've worked with folks so far in putting together a conjugation kit for these labels, something which I'll describe in detail. We've worked with a partner to come up with a very easy conjugation kit to do this, right? It's an overnight incubation. And the way I like to think about this is, look, if you have a conjugation kit tomorrow, you can be asking more questions per cell and having to do easier data analysis and plug and play these into current panels. And I think it's topical too, because we know there's a lot of experimental planning going on right now with limited access to laboratories. So I think it's an exciting time to think about how these could be leveraged in future experiments. Oh, and then just one more point here. We're relying on oligonucleotide manufacturing, right? Which not only we're doing at scale for the current 6 labels I'll show you, but it's incredibly reproducible, right? So like our spec for lot to lot consistency is really incredible, and we QC everything we ship out of our facility on cells every single time in a flow cytometer, right? So we're trying to meet a bit of a higher bar with respect to quality control for what we can present to a customer with our labels. So the first capability then we want to really prove out is this notion of tunable excitation, right, and emission. And so these are the first three labels off the blue, and then we've got 3 off the yellow green. All 6 are designed to be used simultaneously, right, on a number of different instruments, which is why also we went after these 2 different laser lines. And you can see that we're getting excitation, right, and we're actually going to separate that excitation from the yellow green. And in this case, we're able to step the soak shifts out of those labels. And if I overlay these blue and these yellow green labels, you can actually see again that we've separated the blue and the yellow green excitation so that you can use these laser lines independently, right? And we'll talk a little bit around performance characteristic in a moment because we've got some data on that to see how they stack up in terms of brightness. But you can get a sense of what we're trying to do here, which is stack each of these laser lines very, very cleanly to use them independently. So we took these spectra, we published them up on this open source resource called FPBase, which is a really nice resource for looking at spectra, right? And it's free. You can also compare the labels if you're using experiments, for instance, where you want to use this in GFP or RFP or any of the other labels. All the data is in there. So a really nice resource. And then we're very excited that Cytek took our spectra and put them up on their viewer as well, and really enables people to think about how they could leverage this to really go higher on their plex, something we'll describe in a moment based on some of the really nice work that they've done up in the 30 and 40 color range. But I think what's nice about this plot as well is it gives you a sense of what we're doing, right? Which is that, incredibly clean fluorescence, really hitting detectors independently. And then, honestly also gives you a bit of a hint in terms of what we're up to in the future, right? Where we're stacking these laser lines. Because, I think one of the other pieces of this plot that people have been very excited about is they would say, well, tell me about all the white areas. And I say, that's an emerging story for us, right? And so, I think we've got these current 6 labels that I'll show you. I'll show you the performance characteristics of these. We've actually now, the update would be that we're proving out the ability and actually building and doing this at manufacturing scale to do the highest brightness across all of these. So doing a 4 cluster label, which is what I'll explain in a moment, across all 6 of these labels, and then of course expanding our color capabilities, which is very exciting to see. So the first part of this though is like, well, how do they compare to other stuff that I'm using, other dyes that I'm using? And so we took 2 of our labels, and they've got the other 4 that they're performing similar experiments on now, and got this to Mario Roeder's lab at the NIH and said, do your worst, right? So show us separation index, look at MFIs, how do these compare on brightness? How would these be characterized? How would you think about slotting these into your panels? And so to compare them, they really threw the kitchen sink against the NovaYellow dye, right? Looking at it on a CD4 characterization basis, right? Which, for what it's worth, is just a great way of looking at how you can design panels, what the different dye characteristics are when you're thinking around panel design. Something that I was an advocate for 2 years ago when we were really pushing a CD4 kit when I was working for my previous company, and figuring out how could you do this and really learn how to design the best panel for your instrument. And so that's a similar approach that they took with CD4 and NovaYellow, and then CD8 was conjugated NovaBlue, and really thinking around an apples to apples comparison with this Brilliant Blue 630. And this is a 5 laser, it's a symphony instrument, right? And this is run on beads and PBMCs. Although what I'll share is that all 6 of these labels have been were developed on the Attune, which is what we have access to. So it doesn't require any special laser power to do any of this. And so it is still a very nice apples to apples comparison to see this on the Symphony. And I'll show you some data off of the Attune as well for these 2 cluster labels, because that's what this is. These are labels that have the component dyes on 2 of the 4 arms of that cruciform structure. And so the first comparison that we'll do here, just to orient you, is we've got 5 detectors off the blue, 8 off the violet, 7 off the UV, 3 off the red, and 5 off the green. And we're looking at these histograms as a comparison between, in this case, Brilliant Blue 630, which is in this dark blue color, and NovaBlue 610, which is in this light blue color. And you can see that in the primary detector, it's not as bright, right? We'll talk about, again, we'll come back to this notion of what is brightness in a minute. But there's a cost of doing business for being that bright for this Brilliant Blue dye, which is that it's incredibly fluorescent in the violet, right? And this is something that we've seen with a lot of the Brilliant Blue dyes, which you can expect, honestly, based on their, sort of their parentage. But notably, all of our dyes, all of these labels are very, very clean in the violet, right? And incredibly clean in the UV as well. And so, you're able to think about this as a independent blue dye, right? One that you would be able to leverage and then actually open up a lot of the things that you're trying to do in the violet without the compromise of spread. The other one that was really, that honestly became a star dye for us is this NovaYellow 610, right? So what we did is compare this, in this case, to Alexa 694, PECF 594, PE Dazzle, right? An incredibly bright dye. And then this ECD dye from Beckman. And we said, well, how does this label now stack up? And what we found is in the primary detector, it's as bright or brighter than each of these commercially available fluors. But notably, it's also a lot cleaner, particularly in the violet, as you can see across all of these different plots, right? So you could open up the violet. And what's very interesting is in the blue, at least in the comparison to PE Dazzle and ECD, right? So it's a very interesting, it's actually our highest separation index dye when we compare between highest stain index dye when we compare between all these 6 labels. I'll show you the separation index data in a second, but it is incredibly bright, right? And this is in its 2 cluster version. And so this was very exciting to see, because we know we had something that also could add quite a bit of separation power and could be a very, very bright dye, which we could tune up to be even brighter. But the other component of this that I think is the inverse of this, because normally when we expect to see is, okay, you have a very high bright dye, and so that means I've got to deal with some compromises on spectral spreading, and quite the inverse is true here, right? So when we got these these results back, Steve actually shared with us, well, actually this is actually the lowest spread we've seen versus all the other dyes that we measured, right? Which is kind of an interesting result, because now you have a dye that's commercially as bright or brighter than things that you're using off of the yellow now, but do not spread, right? So it's a very interesting dye. Think about sort of almost like this uniform, if you will, peak that's just hitting that detector. And NovaBlue, similarly, in an apples-to-apples comparison with Brilliant Blue, has a very good spread profile, right? And so I thought this was pretty interesting. We'll bring both of these labels back when we think about brightness tuning. But the other result that I think is equally as important when we think around the notion of brightness, right? Because we've never sort of categorically defined brightness in our field. What is brightness equals what, right? It's hard to really know, right? Because you can tune laser power, you can change your gain, right? I think in our, when we're thinking about it, brightness really means the ability to separate populations, right? So can you separate populations that are high or mid or low expressing? And in this case, the simple test is, can you separate CD4 and CD8 T cells? And what does the difference in between the positive and negative populations actually look like, right? So what does that look like? And how is that relative, for instance, to the spread that's in the negative population, right? Because if you have more background, you have higher spread, right? You might end up with the same delta in between the positive and negative, but you'd really like to ding yourself if you have high background, right? And so in this case, we can actually see that NovaBlue 610 and NovaYellow 610 actually have the same separation index as APC. And so we can really hang our hat around saying, look, the ability to do biology in terms of the brightness, these two cluster labels really are, they're ready to be used in current panels to begin stacking the yellow-green, blue and yellow-green lasers really on any instrument. And so I jokingly made this lab mate plot, which I originally called the roommate plot, which is, when you choose a roommate or a lab mate, you really want them to not be dim but clean, right? Because you really want them to help you with your homework or your lab work. You really want them to be bright and clean. And if we were to add some axes to this, we would have spillover on the x-axis and separation index, bright or brightness on this y. And if you really think about it, this is really important, because this is thinking beyond just the notion of bright. What this is really saying is this data will be easy to analyze and interpret, right? It'll be, when we think around leveraging any kind of interesting algorithms in this, or even just performing manual gating, right? Something that I know a little bit about. They really fit. That's really where that fits. Easy to analyze data, right? Easy to interpret. So NovaBlue is dim, but it's incredibly clean. And this is dim relative to what's out there. But as I'll show you, you can actually tune the brightness up on that. Regardless, it can be used together, with a set of fluorophores. Whereas NovaYellow 610 really fits in as both bright and clean, component, right? So, transitioning away from okay, what does that dye performance look like, and into more around consistency and stability. And so stability, so we had a lot of pushback, honestly, not pushback, but I think good feedback when we said we've got a whole new set of fluorescent labels. And people said, okay, that's great. We understand the story around this blue, yellow, green and the stacking. We're interested in that, but how stable are they? Can they be used together? Do I need any special buffers? Right, so this whole set of evolving questions around what is this going to look like, right? And a lot of that came from really two different groups. I say three different groups, right? First was in academia, which said, what happens if these get left out on a bench? Which, I've probably been guilty of that at least once. The second one was in high throughput studies, right? So people shared with us some data that was like, look, over 8 or even 12 hours or longer when I'm running an experiment, I see differences in the performance of a dye, which is interesting. We'll tackle that in a moment. And then the third one was really, people who are running longitudinal studies, right? And really wanting to say, look, how consistent is this over time? What does that mean in terms of stability? What does this mean in terms of fixation? And can I run things later, right? Something that becomes obviously an acute issue when we've got limited access to instruments. So for this first test, probably the simplest test we could do, which was, we literally shipped product across the country and then back, right? 3-day shipping without ice. We register over 100 degrees on the shipping, and we get only this 9% decrease off the spec, which we're reading, but really no change in the spectral profile, right? But I think what's even more interesting about this, because there's this, and we're actually shipping on ice now anyway, is actually what we found when we left these at room temperature. Quite possibly the most boring experiment in terms of executing maybe ever done. Because you just literally take a vial and leave it under tinfoil on a bench and you just wait, right? But sometimes maybe it's those simple experiments that honestly give you some of the most interesting data, right? And so we've actually tested this out to 3 weeks now, and there's no difference. That's just the longest we've tested. There's no difference, right? So you can literally leave these on a bench. So if they're room temperature, they're incredibly stable. And for what it's worth, some of the reasoning there, and this is a question that probably is just worth getting ahead of, is that phyton structure, that cruciform structure, if you were to lay it flat, it'd be 20 nanometers across. It actually has some curvature to it, right? So it's probably about 19 nanometers, maybe 18, and in the high 18s, if you were to measure it, the width of that. And what's interesting about that is, because it's a DNA platform, the nature of it being DNA solubilizes and really protects the dye-dye interactions, right? You think about those dyes not being incredibly labile once they're on the DNA platform, because they're really fixed. So we've seen some interesting results on this. We're testing some of the photo stability right now, but you can see that the physical stability of these is really interesting. So let's push that, right? So let's push that physical stability. How stable are these things, right? So we did really, I think, what is an interesting experiment and honestly chemically abusive to fluors, right? So how could you do that kind of experiment when you really push that physical stability? So what we did is we took, fresh PBMCs and stained them with our NovaL610 dye that was conjugated to CD3. Okay, and so this is the result you get. This is often a tune. There's nothing, no symphony, no high-end instrument being leveraged for all the experiments that I'll show you for the rest of the time, except when I clearly call it out, right? And what we did is we took that, and then we also took cells that were stained with the same antibody, NovaL610 conjugated CD3, that were fixed with 1% paraformaldehyde solution and left for 2 weeks, right? Tinfoil in the fridge, right? This is like the lazy student experiment, right? Where you just say, oh, I'm just going on, I'm going on vacation, or I got to work from home for 2 weeks. Let's fix it, right? Turns out there's no difference. And, kind of an interesting and honestly somewhat incredible result, right? When we think about having limited access to laboratories, this suddenly becomes incredibly interesting. But it's also interesting from a chemistry point of view. If you think about leaving something in 1% PFA for 2 weeks, right, which is just the longest we've tested, that is an incredibly intense solution to be leaving fluorophores in, right? And so, I'd love to see us expand that. I'd love to see us sort of put out the challenge of like, put it in this, pretty aggressive solution. Let's see how fluorophores stack up. And I think I'd love to see us get to that soon. Okay, so the third part is this notion around tunable brightness, right? And how can we go from 1 cluster, 2 cluster, up even to a 4 cluster label? And then what would that mean for panel design, right? Which is something we're still trying to wrap our head around, although we'll take a pause in terms of thinking around how are we working with customers to investigate how this could enable some new biology. For what it's worth, everything I've shown you to date, some of the NIH testing data, all of that was done in 2 cluster. And so you can get a sense of the performance of that. It's also the way our conjugation kit works for now, although I mentioned we've got a conjugation kit that we're opening up for people who are investigating new capabilities using this 4 cluster label. So the first part of this was, can we replicate networks, right? And this is just like the good science experiment, because if you think about this phyton structure being essentially symmetric along this y=x axis, along this diagonal, you really want to prove out that you can replicate it, right? And so you can replicate these networks. For what it's worth, there's also some interesting things. If you remember, I talked about earlier that you can actually look at the square of distance, right? Remember, when you're doing the sequence design, you can have very, very small differences in that distance. And you can actually see there is a tiny difference actually. So you can actually get less, less of this emission that's not desired, and you can really try and even hit that more cleanly if you're just doing, for instance, a 1 cluster. But that's getting down into the weeds, right? Really where we want to be is, now that you can replicate these networks, now you can create a 2 cluster label, and you can think about tiling this and creating a 4 cluster. And it's this replication that enables tunable brightness. And so you can actually see here, you've got the blue that's a 1 cluster dye, and this orange-red color that's a 2 cluster. You can actually see it's in fact twice as bright. And of course, a spec plot's one thing, but we all want to see the flow, right? That's really where it matters. And so that's exactly what I'm showing you here. And this is off of this NovaBlue label. So this is a NovaBlue, which has a 1 cluster just off one of the arms, and then NovaBlue that has a 2 cluster label. And so the first thing is the fluorescence -1, right? This is just the cells, no phyton, no antibody, no nothing, just so we could set our gating correctly. And then we've got the label only. And so in this case, what we've done is we've doped in just the phyton, right? Either the 1 cluster or the 2 cluster label, just to get a sense of what is the background that's being added here, and it's incredibly low, right? And then what we can do is then compare the CD3 that's been conjugated to a 1 cluster NovaBlue, and that one that's been conjugated to a 2 cluster NovaBlue. And you can in fact see here that it is in fact twice as bright, right? So we're looking at this on a bi-exponential scale, where it's logarithmic, really out past probably 10 to the 2 in this case. And you can actually see it's twice as bright. But there's another thing to notice about this figure, and it's something that struck me when we were looking at this data again, right, a little while ago, which is that we haven't changed the standard deviation in negative. Neither have we changed the standard deviation in the positive, right? On an absolute basis. And so while we're getting brighter, we're not changing we're not changing the brightness performance of that label, right? It's still maintaining its tight population. So that it still is facilitating the notion of this easy data analysis, right? So I actually am increasing, if you look at this, the staining index, right? The ability to actually separate the separation index, the ability to actually separate these populations. And I think that's really interesting when we think about, I've got this low or mid expressing population, and then it's saying, okay, let's increase the separation index of that. but let's do it without compromise. So how do we do that? So let's push that even harder. So what we did is we took our NovaFluor 610, labeled this one that's got a really incredible performance off the yellow-green laser, and said, let's push that hard. Right, so we went in and created this 4-cluster label. You can see that it's actually twice as bright here. For what it's worth, if you actually normalize these, this is more for, this is for the fluorescence geeks in the room rather than the flow geeks. Maybe it's the same now, I don't know. You actually get less spillover off of the 4-cluster, right? And that's because you have some labels where you're getting even more efficient FRET off some of the clusters. So for what it's worth here, this is this Nova yellow, right? And now what we're doing is comparing the 2-cluster to the 4-cluster, right? And you can see, in fact, it is twice as bright. Because I see some of the names that are on this, and I know you guys are smart enough to look at this and also ask me the question, I'll try and get ahead of it, which is, Mike, why do you have such high background off the 4-cluster, right? And the answer here is actually that this was not treated with the blocking solution that we developed for monocytes and macrophages, right? Very similar to the BioLegend TrueStain monocyte blocker, quite a bit of a different formulation, but non-protein, non-antibody based. And it's because some of our Nova dyes, very similar to the Cy5s or Cy7 conjugate dyes, or if you're using them on your own, bind onto monocytes and macrophages. So we went in and did the work of receptor characterization there and designing that non-protein, non-antibody based solution to really block that. So that's in fact not in this solution. So we've got new results that show that you can take that background down to 0. And again, get that same notion of increasing the separation index, right? Without an appreciable increase in the standard deviation of those positive peaks, right? And so what we did is we attempted to say, hey, look, let's compare this to P's brightness on an MFI basis in its primary detector. And so in this case, remember what we've got here is actually a 1 antibody and 1 phyton system, right? And in that system is actually quite quantitative, right? Because we know exactly how much dye is on it. We know there's 1 phyton, 1 antibody. In this case, we have 1 4-cluster and 1 2-cluster on 1 antibody. And so you can see we do get, again, twice as bright on an MFI basis, right? When we think about this 4-cluster. But I think what's interesting to look at is looking at how just the really, if you will, the lack of spill into the adjacent channel, and then essentially not much, if anything, going into the blue, right? And so this is very intriguing because you have the ability to hit that very, very cleanly, but you can still use the adjacent channels. You could still use your blue detectors. P, where there's multiple on antibody, really it's a distribution of 1 to 2. You get incredibly high brightness, right, in an MFI basis off the primary detector, and you destroy, right, the ability to do some good analysis off of the adjacent, and certainly you spill that directly into the blue, right? So on a flow basis, we've shown that it's 1.84 times as bright, nearly twice as bright as the 2-cluster. We say it exhibits P-like brightness in terms of its ability to actually separate populations. But we've also got the ability to actually adjust the degree of labeling. So we could have multiple 4-cluster phitons to 1 antibody, and then tune that up even higher. But of course, one of the key things here is it exhibits a heck of a lot less cross excitation than PE. So the way we think about this is actually the ability to use this across all of the different colors that we've talked about, all 6 of those labels. And so that's actually what's going on. And so we've got our 4-cluster NovaFluor 610, which is currently in beta, and we're also deploying all 6 of our labels actually across that 4-cluster. And here's some things that we're thinking about, right, and things that we're working with customers on. So the first one is small particle and extracellular vesicle detection, right? And so this is, can we trigger on fluorescence using a very, very clean and very, very bright label where we can actually, put antibodies onto an EV, trigger on that, and detect that. Quite interesting work that's being done there. PE and PE conjugate replacement for undetectable drug targets. So we're working with some folks who are currently using PE or PE conjugate or another commercially available label, and they're saying, look, we'd really like to get to the point where we've got something where we can go potentially much, much brighter if we go to a high degree of labeling, but certainly much, much cleaner so that we can use that in multiplex capacity in our assays with the stability characteristics that you guys can bring to bear, right? That's been some exciting work to see. The other one is we're doing internally is some limited detection testing, right? So putting these on essentially beads and saying, okay, how low can we go in terms of what the level of expression on a cell might be in sort of a simulation mode to say, okay, how low could we possibly go? There's also been a lot of interest in sort of, hey, how can we look at simultaneous checkpoint inhibitor detection, right? Can we detect multiple low antigen densities? And can we even detect those, for instance, earlier in our clinical trial? So sort of a smattering of things that the customers have been interested in and are aiming to deploy this on. So very, very exciting. And I'd also say, an area that is certainly an update versus even where we were a couple weeks ago. This is what I mentioned earlier, which is we've got these two different brightness strategies, but I'll use this also to talk around the conjugation kit, because I think it's interesting, and that's just kind of where we are for now, right? So two different brightness strategies are, we can tune up the clusters, which is what I've shown you, right? Which is how do we leverage the platform to do that. Again, we can do that in a one-to-one basis, and there's some interesting notes here that we could get into maybe in the Q&A session and just sort of geek out about sequence-tagged antibodies for a little while, because you know I'm totally game to do that. And then, but the other one is this notion of degree of labeling, where you can actually change this degree of labeling, right? And so what we've actually found for the conjugation kit is the best degree of labeling is about 3-to-1, right? And so that seems to really produce antibodies that are really, really optimal in terms of their brightness. And the conjugation kits, so we went out and we vetted a number of these conjugation kits, right? And some were a heck of a lot better than others. And so we ended up partnering with someone who created one that we really liked. It had low hands-on time, and you could be using the antibodies tomorrow. And so what does that look like? So the steps are, you know, you've got this antibody here. We actually, you see there's an activation step. So what you do is you activate the NHS esters on here that are sitting on lysine. So very standard NHS ester chemistry. There's no special chemistry that we're using in there. And then what we can do is actually, you know, we use this partner's kit to take a single-stranded oligo and put it onto the FC region. And I say onto the FC region because they've tested thousands of different antibodies and have never seen it mess up the ability for antibodies to bind. So that binds onto we've got that single-stranded oligo that's hanging off of the FC region, and then you can take our label, which will then specifically bind onto that single-stranded oligo that you've got. And for what it's worth, because you're leveraging this oligo chemistry, you've got this DNA interaction, we've actually seen the efficiency of that's been anywhere from 60 to 80%, right? So which is very, very high. And then there's no post-purification necessary. You can wash the phytons out. And as I showed you, they don't really bind to the cell surface really well when we looked at that just doping phyton experiment, right? So it's, you know, it's very, very low hands-on, low number of steps. And so the way we've done this for our current labels is do that in a 6x100 microgram kit, right? To produce a sufficient amount of antibodies, right? We could do a really good number of tests for that. And so we'll discuss that a little bit more at the end. But really, you know, hopefully I've shown you some data that's proven out this notion around bright and clean, around engineering for bright and clean, consistent and stable, which is really the lift that we get from the physics of the platform. And then this notion of controllable brightness, again, which we're deploying across all the 6 current labels, but then we'll do that on a go forward across all the different labels we put out. And now we're investigating, okay, well, what could be possible if we do have this, if we do have controllable brightness? Just something else that's, I think this is more at this point just for fun, because there's a huge amount of biology that stands between us leveraging spectral labels and thinking about this, but could be interesting for other applications, right? Is we created 4 different spectral dyes, and we did it in 2 weeks. And so that's actually kind of another note in here, which is just the pace of development, right? And so we're hopeful that we can, you know, get through these current times and actually get a large set of labels into researchers' hands really over the next, you know, really over the next year, and show them what's possible. But this gives you a sense of the pace. 4 spectral labels, 2 weeks, and we're able to essentially design spectral labels that we know could be separated, right? And I was having a great discussion last week with someone, you know, back when you could travel, and you know, they were like, how's this going to work, right? And I thought, well, the best analogy actually is cell phones, right? We're doing this all the time, where cell phones are communicating with a tower, and the way they actually communicate is actually using these signatures, right? Very similar types of signatures, and they're deconvoluted. Now, the issue we've got in biology, of course, is you got underlying antigen density, right? That can be variable. And so I think that's what the biology that stands between us and leveraging these fully, but I think it's an interesting as a capability nonetheless. Okay, I'm gonna take a swig of coffee, and then we'll talk about panel design. So I'm not going to describe conventional panel design again, but I do want to talk about paradigmatically, like, let's take a step back and let's figure out, like, what's the goal of actual panel design? Really, you're attempting to even out positive signals. You're trying to increase this chance of positive negative separation. You're dealing with current labels to do that, right? And you're kind of pushing it to the max in terms of the compromises, particularly if you're thinking about something like a PE or APC. What does it miss? Cross-excitation from broadly absorbing labels. I mean, we account for it, but we don't specifically sort of engineer our panels for that. Spillover from noisy labels, I mean, this continues to just be the not magic part of flow cytometry, that, you know, the more that we cram together, right, and we're relying on this underlying dye performance, we do have channels, particularly once we get in a high parameter space, where we're detecting more spread than signal. That's scary, right? It's just, and why is it scary? It's scary because of the difficulty of data analysis and data interpretation, right? And so, you know, we're trying to move to a spot where we can say, look, it's not just that we've got clean fluorescence. It's not just that we can do plug and play, but it makes the data analysis and the data interpretation easier, right? And sort of proving that out. And so there's a project that we've got now to take that on. So we try to take a bit of a lateral view on this, which is, well, okay, what if we design a panel that minimizes cross excitation and spillover? And so we came up with a small parameter example to try and make this work. And so what we did is we took a TBNK kit, a conventional 6-color TBNK with conventional fluorophores, right, which is off the blue and the red laser. It's a test that's been with us for a very, very long time. And for some historical reasons, it was off the blue and the red lasers, right, which is essentially what do we have access to when the field was originally creating a TBNK kit, but now it's Legion, right? And so what we said is, okay, well, how would we do this how would we do this and challenge ourselves to do it? How can we do it off of the adjacent blue and yellow green lasers using the clean 6 labels that I've shown you? And so, and again, it was an exercise in like, can we challenge it by pushing the yellow-green stacking? Can we challenge also the ability, can we challenge the thinking that we've had around panel design? And so this is actually just a good result to look at anyway, because this is actually just looking at what does each of these labels look like, right? And this is just run on a tune. Again, this very standard configuration here. You can see for what it's worth in the primary detectors in pink. Nova Blue 530 is this another almost fantasy dye, right? It only emits in blue 1, which is pretty amazing, right? And so the spectral profile of that dye is fantastic. But we looked at all of the different fluorophores that we've got here, and you can measure, of course, the MFI of not only what's going on in the primary detector, but also in all secondary detectors, right? So you can extract that. The other thing you can do is it's simply essentially as an abstraction of what the actual antigen density is, because if you have real antigen density data, you sort of have the keys to the kingdom, right? Which is we just looked at the MFIs as a proxy of that antigen density across all 6 of this. And the way we did that is simply by using the same label, right, across all of these different markers. And what we could do then is actually permute all of the potential panels that we could make, right? We've got our 6 labels, we've got some antibodies, we've got to ask, let's permute every single combination of every single antibody, every single fluor combination we could do, right? And so what we could do then is measure that, right? And so the way we would measure that is we could take the sum of the off-channel fluorescence, right? So we're actually dinging ourselves, right, if there's off-channel fluorescence at all. And we can optimize for that in a way that we're really not doing now in panel design. And then we can also take the sum of the primary channel fluorescence. And we can think about this now as an optimization problem, right? Not a let's deal with compromises problem. And so I think in my inimitable fashion, I had the label, the quadrants here, but what we really want to optimize for is low off-channel, high primary channel. So really you want to end up in the epic amazing box. You don't really want to end up in serviceable or in these other boxes. And so what's really interesting for what it's worth, and I was somewhat blown away when we first saw these results, is that panels fall into clusters themselves, right? And so this isn't data. What this really is it is the mapping of the total off-channel fluorescence and primary channel fluorescence, and panels cluster. So if you take the conventional method where you take the highest brightness and map it to the lowest antigen density, you end up with not a very good panel, right? You end up just with a serviceable panel. But if you optimize for the total off-channel fluorescence and total primary, you end up here, right? You end up in a space where you are optimized, right? So it's actually quite interesting from a how are we thinking around panel design generally point of view. So again, here we've got, in this case, the 6 off of the yellow and the red. Now we're going to try and 6 off the adjacent. And so so here are the flow results. We can gate on LIMS here, very obvious, then CD3 positive. We're obviously getting some degradation of the P size 7, right? This is commercially ordered and fresh, from the vendor. So that's pretty nasty little compensation issue. And then, really where it's going to get pushed, of course, is CD16 and 56, right? Because we're up against PE, which is actually able to pull apart, these populations and do it reasonably well. LIMS, obviously, that's a pretty easy one because it's on it's on CD45 and scatter, so we're able to pull that apart. CD3 arguably should be a little bit brighter, right? We're able to do a very, very good job on the CD8 and CD4 using actually, remember, in this case, adjacent laser lines. And obviously, in this case, while the 19 separation is good, the CD16 and 56 is not the kind of performance that we want to see, right? And so in this case, you'd really want to say, okay, what I really want here is a NovaFluor 664 cluster so that you can then step that out and detect the mid populations. But I thought it was an interesting example of how you could do panel design, how you could do that, and then do that off adjacent lasers and detectors. Okay, so how are we pushing forward in testing NovaFluor dye performance? So been some beautiful work from Cytek in looking at this 35 color panel, right, which they've published, which is great, right, showing this, here's all the specificities and dyes that we're using. And so, we looked at this and thought, wow, wouldn't it be great if we could take some of this great work and then actually drop in our 6 labels, right? Could we sort of immediately add that clean fluorescence and go higher on plex? And so that's exactly what we're up to. It's exciting. We're actually finally analyzing this data now. And so hoping to share that very soon. But this is what we did. We dropped in an additional 6 antigens here, right? Again, off the blue and the yellow. And one of the reasons that we can do this, again, is because you're just essentially plugging and playing these clean fluorescent labels into the panel, not adding appreciably to spread, right? Which we hope will make the data analysis easier, right? Something that we have to prove out. So to give you a sense of that, although this is a very busy plot, this is what we did, right? We dropped in Nova Blue 530, 610, 660, and the Nova Yellow 570, 610, and 660 directly into that, into the 34, it was 34 commercially available labels, and then dropped in these 6, right? And so, this is really our push to go above and beyond 40, and then take this data and have it be really a resource for the field. So I'm optimistic that we'll have more news on that very, very soon. But that's the update. That's the update for now. So hopefully, I've shown you these 3 different things, which I'm not going to beat into the ground, but hopefully also a little bit of a different way we can think around panel design and how we use our instruments. And then a final note, and then we'll have plenty of time for questions, which is how are we wrapping all this up, right? How are we working with customers now? And we can even talk about maybe in the Q&A session about what that'll look like in the future. The first one, which is something that we've already been shipping, we started shipping them out on Valentine's Day. Had a great note from a customer saying this is the best Valentine's Day present I've ever gotten, which is, that's pretty awesome, honestly. That's a lot of love. Is this NovaKit CD4 test kit. And so what this is human anti-CD4, the SK3 clone, which, I think we're all very familiar with. It's an old friend, conjugated to the 6 current NovaFluor labels, right? And this enables folks to assess these labels on their instrument of any kind, right? And then say, okay, how would I work these into my current panels? Then there's the NovaFluor conjugation kit. It has the 6 NovaFluor labels with NovaBlock. For what it's worth, let's spend a moment on the blocking. The block step is, you can do it with FC blocking. You can actually do it as part of your stain, right? It's sort of interchangeable with your, I shouldn't say interchangeable. It is implementable as part of your standard flow cytometry workflow. This conjugation kit enables you to conjugate 6 different antibodies at 100 micrograms a piece to all 6 of the labels. So it's a very enabling conjugation kit in the sense of its volume. And then we've also, honestly been pretty surprised, but excited around the ability to also work with people on custom conjugations, right? So saying, look, I really want, this CD marker attached to Nova Yellow 570 because that hits a channel across a number of my panels. And so we've worked with folks on that as well. And then a final note is, we are deploying this beta program, which is something we really want to bring, folks closer to as we do new colors or new brightnesses. And we're doing a 6x10 microgram conjugation kit for the Fluor cluster labels, right? If there's some interesting biology that we say, okay, like this is worth collaborating on, let's think about that. For folks who are already looking at the, what we're doing now across all of these 6 colors. So with that, that's not my email, that's Stedin's email. But my email is mikestadnisky@Phitonex.com, and I'd be happy to take any questions. If I can get back to Zoom here. There we go. All right, still no open questions, so I will hang out for another couple minutes, and if and when the questions start firing, let me know. And if not, My email address is actually up here at the top, or at least it used to be. I'll just type it out for you so you have it, and then you can email me any questions. All right, cool. See, no questions. Just to let you know, we do post up all of our webinars up on our YouTube channel as well. So if there's something you missed or want to replay, we'll be posting that up within a couple of days. So thanks everyone for attending. Hey, here we go. Sweet.
I'm listening.
Oh, Siri's listening. Okay, good. Not quite sure why. So question, could you please elaborate on the 4 colors mix? 4 colors mix. Could you clarify that question? I'm trying to think around what you mean by 4 colors mix. You're talking about the 4 cluster, which is very, very bright. Yeah, I'd love to learn more about what you're curious about there. Slide 36. Let's do it. That's really impressive that you remembered it. Ah, yes. Got it. So yeah, this is interesting. So what we did for this work is take component organic dyes that we knew that worked on platform, and we created a 1 cluster spectral dye, or sorry, 1 cluster, 4 different 1 cluster spectral dye, right? So you can see here, this is cartoonized here, but it gives you a sense of the kind of fluorophore networks that we could create to create a spectral dye. This is the measurement that you get out when you measure it on the spec, right? Because we don't have access to, for instance, like, you know, a Sony instrument or something where we could do sort of this, you know, or even at this point we didn't have an Aurora, we didn't have any ion instrumentation access at all, and so you get this mix, and you can perform linear least squares, right, to determine, you know, what are the component labels in this mixture. But the nice thing about this is, if you think about it from kind of our current paradigm, is we know what the signatures are going in, right? So if you had the, you know, a library, for instance, of these component signatures, you should be able to unmix them. The other point I would make here is, and you know, I think this is something that's interesting to test, and we just haven't tested it yet, is what's the impact of underlying antigen density, for instance, on detecting these different signatures, and then the ability to deconvolute them. I think that's kind of interesting to consider, right? For what it's worth, anytime you perform LLS, because it's an overdetermined system, it's an overdetermined equation, you can choose what to optimize for. So if you had some underlying concept of like what the antigen densities would be, you could actually probably optimize for, you know, the different underlying biology here, expecting to detect more or less of these different signatures. That's purely me just thinking about what's possible versus any experiments that we've done, which that's an important kind of distinction, right? I always try to say, look, we haven't tested it yet. And really what we've done is make these at R&D scale. So hopefully that answers your question about that and gives you a little bit more color on what we're doing. All right, I think clearly Zoom needs to implement someone is staring at the computer, seeing if there's any questions left, music as part of that. So we'll have to file that feature request. So with that, I'll stop share here. Again, feel free to hit me up with any questions. Thank you guys so much, and we'll post the recording soon.